Welcome to this video on practical disease concepts in epidemiology, based on Chapter 3 of Introduction to Epidemiology by Ray M. Merrill. In this lesson, we'll break down how diseases are classified, transmitted, and tracked. You'll learn about incubation periods, disease stages, immunity, and public health tools like isolation, quarantine, and herd immunity. Let's explore how epidemiology translates disease biology into public health action. Disease categories. First, let's talk about communicable and non-communicable diseases. Communicable diseases are infectious and transmissible, like measles or hepatitis. Non-communicable diseases are not spread person to person. Examples include diabetes, cancer, and heart disease. Diseases can also be classified by cause. Congenital or hereditary, for example, Down syndrome. Allergic or inflammatory. Generative, for example, Alzheimer's. Metabolic, for example, diabetes. Cancer. This classification enables public health professionals to organize prevention and treatment strategies effectively. Etiology and transmission. Etiology is the study of the cause of disease. A key distinction: all communicable diseases are infectious, but not all infectious diseases are communicable. Or, for example, tetanus is infectious but not passed from person to person. There are two major forms of transmission: vertical, from parent to child; horizontal, from one person to another. Common transmission routes include airborne, waterborne, vector-borne. Direct contact, pathogens, and host responses. Pathogens are organisms like bacteria, viruses, and parasites that cause disease. Key terms: infectivity, how easily the pathogen spreads; virulence, the severity of the disease it causes; toxins, substances that damage host tissues. The interaction between pathogen and host is influenced by genetics, immune status, behavior. Environment. Understanding these factors helps us predict outbreaks and target interventions. Disease progression. Diseases often follow a timeline. First is stage of susceptibility, before exposure. Second is presymptomatic stage. Pathogen is present but no symptoms. Third is clinical stage. Symptoms begin. Fourth is recovery, disability, or death. Important periods include incubation period, time from exposure to symptom onset, for example, 1-3 days for flu. Latent period, time before a disease becomes infectious. Infectious period, when a person can transmit the disease. Knowing the incubation period helps determine quarantine lengths. For example, Ebola is 21 days. Isolation, quarantine, and public health tools. To protect communities, public health uses isolation for those confirmed to be sick; quarantine for those exposed but not sick. Example, Nurse Cathy Hickox, quarantined in 2014 after returning from West Africa despite being asymptomatic. Her case raised important legal and ethical questions. Other tools include case reporting for notifiable diseases, for example, TB and COVID-19. Zoonosis, control diseases spread from animals to humans, for example, rabies. Immunity and herd protection. Immunization helps build two types of immunity: active, developed from infection or vaccination; passive, acquired, for example, from mother to baby or through antibody injections. When a large portion of a population is immune, it creates herd immunity, protecting even those who aren't vaccinated. This is critical for diseases like measles. Environmental and host-based control strategies target: environment, ensure clean air, water, and sanitation; host, promote personal hygiene, nutrition, and vaccination. We also address malnutrition, both undernutrition and overnutrition, and chronic conditions like obesity and disability, which affect health outcomes long after disease onset. Disability classification includes impairments, activity limitations, participation restrictions. Let's summarize what we covered. Diseases are classified by cause, transmission, and progression. Public health uses terms like incubation, latency, and infectious period to plan interventions. Isolation and quarantine remain powerful tools. Immunization and herd immunity protect populations. Reporting, sanitation, and nutrition play key roles in disease prevention. Understanding how diseases develop, spread, and can be stopped is essential for effective epidemiologic practice. Join us next time for Chapter 4, Descriptive Epidemiology and Study Design.